Navigating the genetic maze. What is passed down? Welcome to the maze of genetics. Whether you're exploring the journey of your own DNA or learning about the pathways that shape who you are, it's important to understand that genetics plays a big role in our lives, but it's not the whole story. Some of the changes we encounter are inherited, and some are new, happening spontaneously. As we navigate the twists and turns of this genetic maze, let's take a closer look at the paths we may encounter. Your genetic journey begins at the very start, when the sperm from your father and the egg from your mother come together, marking the entry point of your life. Think of the sperm and egg as two explorers, each carrying half of a map that will guide your path. Neither has the full picture, but when they combine their pieces, they create a complete genetic blueprint for you, the new individual. Within the sperm and the egg is the genetic information from each parent. When they join, they create a set of unique genetic information, which is the basis of a new life. The egg from your mother provides half of your genetic information, and the sperm from your father provides the other half. This genetic information is packaged in structures called chromosomes, which contain the instructions that shape who you are. The egg contributes 23 chromosomes, and the sperm adds another 23, combining to form a complete set of 46 chromosomes that make up your full genetic blueprint. Here you can see the 23 pairs of chromosomes. One of the first factors that shape a new individual's identity is their sex, determined by the sex chromosomes, the X and Y chromosomes. These we inherit from both our mother and our father. Think of the chromosomes as pieces of the map. The mother can only pass on an X chromosome, while the father can pass on either an X or a Y chromosome. If the father passes on an X chromosome, the individual will inherit two X chromosomes, leaving them XX, and typically develop as female. If the father passes on a Y chromosome, the individual will inherit one X and one Y chromosome, XY, and typically develop as male. In this way, the father's contribution determines the final path on the map, guiding the journey toward either a male or a female outcome. Now that we know how chromosomes are passed down, let's take a closer look at how genetic information is stored. Inside the cells of your body, which are the basic building blocks of all living organisms, chromosomes house the genetic material known as DNA, deoxyribonucleic acid. DNA is a long, coiled molecule composed of genes that contains the instructions for your growth, development, and how your body functions. We can think of the cells as the farm where everything is produced, stored, and managed. On this farm, the chromosomes are like filing cabinets that organize and protect the maps, the DNA. The DNA itself contains detailed instructions for your body's development and function, much like a maze map guiding your journey. These maps are made up of genes, which represent the individual paths in the maze, each directing a specific part of the journey. Now, as we dive deeper, we see that DNA is tightly packed and organized into larger structures called chromosomes. These chromosomes safeguard the genetic blueprint, which is the DNA, ensuring that it's kept safe and in order, all within the cells of our body. This organization allows the cell to access the right instructions at the right time, ensuring everything runs smoothly. As you see here, DNA is a long coiled molecule made up of genes. Genes are each responsible for a different function in our body. They're different sections of DNA that contain specific instructions for creating proteins, and these proteins perform many essential roles within our body. In addition to the sex chromosomes, each parent contributes unique genetic traits, such as eye color, hair color, or even the possibility of certain conditions or diseases. These traits are spread across different chromosomes, coming together to shape your individual journey through life. Each gene in your DNA comes in different forms, known as alleles. Think of an allele as a fork in the maze, each option offering a slightly different route, but still leading to a similar endpoint. For example, a gene might control eye color, and the alleles for that gene could determine whether you have blue or brown eyes. So while a gene is the overall instruction, alleles are the variations that provide different versions of that instruction, influencing how traits are expressed and passed down. In our mutations video, we talk about two types of cells in the body, germline and somatic, and two places where genetic material is stored, the nucleus and the mitochondria. This is important for inheritance because germline mutations, which are in the sperm or egg cells, can be passed on to children, while somatic mutations in our other body cells are not inherited or passed on. Mutations in nuclear DNA affect traits passed down from both parents, while mutations in mitochondrial DNA are only passed from mother to child. Knowing these differences helps us understand how genetic traits and conditions are inherited. So now that we have covered that we have genetic information that is inherited from our parents, each generation contributes a new set of genetic information or instructions, shaping the pathways we follow as we grow and develop. But inheritance isn't just about passing on the same paths. It's also about how traits or conditions, anything from eye color to genetic disorders, are passed down through families. The different ways of passing down traits and genetic information are known as inheritance patterns. Inheritance patterns are the rules that determine which traits or conditions get passed on and how likely they are to show up in future generations. Since our genes come in pairs, one from our mother and one from our father, and each gene can come in different forms of alleles, these alleles can either be dominant or recessive. In terms of mutations, a dominant allele is an affected gene that expresses its trait even if only one copy is present. In contrast, a recessive allele is an affected gene that requires two copies, one from each parent or spontaneously, to show its effect. In dominant inheritance, the gene or the allele that carries the condition will overpower any other recessive gene or allele that it pairs with. So if you inherit the dominant gene and allele from one parent, whether it's from the mother or father, that gene will determine if the condition or trait is expressed, even if the other parent passes down a normal or unaffected gene or allele. Dominant inheritance is like clear, prominent pathways in the maze that are easy to follow. You only need one copy of the map carrying the trait to be able to follow this path. Through inheritance, when one of the parents has a dominant trait or condition, there's a 50% chance the condition or trait will be inherited. Let's break this down. The parent with a dominant condition has two alleles, one dominant affected allele, affected for the condition, and one normal unaffected allele for the healthy gene. The parent will pass one of these alleles to their child. The other parent who does not have the condition will pass on their unaffected allele or their healthy genes because they only have two unaffected alleles or healthy genes to pass on. When it comes time for reproduction, the child will inherit one allele from each parent. There are two possible outcomes because each person has two copies of every gene. One copy is inherited from their mother and the other copy is inherited from their father. When it comes time to pass down genetic information, each parent randomly passes on only one of their two alleles for each gene to their child. If the child inherits the dominant affected allele from the parent with the condition, they will have the condition because dominant genes only need one copy to be expressed. If the child inherits the unaffected allele from the parent with the condition, they will not have the condition and will have two unaffected and healthy copies of the genes. This means that each child of a parent with a dominant genetic condition has a 50% chance of inheriting the dominant allele and therefore the condition, and a 50% chance of inheriting the unaffected allele and thus not having the condition. An example of a common dominant condition is Marfan syndrome, where 3 out of 4 times the condition is inherited from one of the parents. On the other hand, recessive traits are a bit trickier to navigate. To follow a recessive path, you need 2 copies of the gene carrying the trait, one from each parent. Think of this like a hidden or secret path in the maze, one that you can't see unless you've inherited both parts of the map. Even if neither of your parents shows any symptoms of the condition, they could both be carriers of a recessive gene. In this case, both parents carry one copy of the affected gene for the recessive condition, but don't actually have the condition themselves. If both parents pass on their copy of the recessive affected gene, you may find yourself following that hidden path leading to a condition that wasn't visible in the previous generation. For example, conditions like cystic fibrosis or sickle cell anemia are inherited in this way. Both parents might be carriers, unaware that they're passing down a recessive gene, But if their child inherits both copies of the gene, one from each parent, the condition will manifest. In recessive inheritance, both parents must carry a copy of the recessive gene for the condition to be passed down. If you inherit one healthy allele and one recessive affected allele from your parents, you'll be a carrier yourself, meaning you won't typically show symptoms of the condition, but you can still pass it on to your children. However, if you inherit the recessive affected allele from both parents, one coming from each, you will have the condition because two copies of the affected allele and gene are required for the condition to be present. Let's break this down. If both parents are carriers, each has one healthy allele and one recessive affected allele. When they have a child, the possible outcomes are 25% chance or 1 in 4 chance that the child will inherit two healthy alleles, one from each parent, and will not have the condition, nor will they be a carrier. There's a 50% chance that the child will inherit one healthy allele and one recessive affected allele, becoming a carrier, but typically not showing symptoms. Lastly, there's a 25% chance or 1 in 4 that the child will inherit two recessive affected alleles, one from each parent, and they will have the condition. So if both parents are carriers, there's a 25% chance that each child will have the condition, a 50% chance that they will become a carrier, and a 25% chance that they will not be affected at all. Genetic carriers are individuals who carry one copy of a recessive gene for a particular condition, but typically don't show any signs or symptoms of the condition themselves. Carriers are like hidden maps in the genetic maze. They possess an altered version of the map, but their journey doesn't show it. This is because they may not realize that they have an altered map relying on their original version, or the original map works well enough that the altered one doesn't affect their path. As we just saw in recessive inheritance, carriers have only one copy of the affected recessive gene or allele, so they can pass it on to their children. The key here is that two carriers are needed to pass down the condition in terms of inheritance. If only one parent carries the gene, the child may inherit the gene, but not develop the condition. The only other possibility for this to occur for the child to develop the condition is if one parent was a carrier and the other gene was affected spontaneously. The challenge with being a carrier is that you may not know it, especially when the condition doesn't affect carriers. They often live their lives without realizing they're carrying an affected gene for a genetic condition. An example of a condition like this is cystic fibrosis, like previously mentioned, where an individual may never know they're carriers since this genetic condition does not present symptoms in carriers. Only those who have both copies of the recessive affected gene. In some cases, a carrier may show symptoms, but they are typically milder than those seen in individuals who inherit both affected copies of the gene. One example of this is hemophilia. Understanding carrier status is important because it helps predict the risk of genetic conditions in future generations. Some paths in the genetic maze are hidden, and carrier screening can uncover these paths, providing insight into whether future children might inherit a recessive condition. While dominant and recessive inheritance patterns are among the most straightforward ways genes are passed down, there are other inheritance patterns that are a bit more complicated and don't always follow the typical rules. These patterns can be influenced by factors like the type of gene involved, whether it's located on sex chromosomes, or even the source of the genetic material itself. Let's take a closer look at some of these more complex inheritance patterns, including X-linked inheritance, mitochondrial inheritance, and others that don't fit neatly into the classic dominant recessive model. These patterns can sometimes make genetic conditions harder to predict, but understanding them is key to fully grasping how traits and diseases are inherited. X-linked inheritance is a specific type of inheritance that refers to traits or conditions caused by genes located on the X chromosome. Since females have two X chromosomes, XX, and males have one X and one Y chromosome, the inheritance pattern differs between the sexes. X-linked dominant inheritance occurs when a dominant gene on the X chromosome causes a condition. Both males and females can inherit the condition, but it is often more severe in males because they only have one X chromosome. For example, Rett syndrome is an X-linked dominant condition that is typically much more severe in males who often do not survive birth or infancy if they inherit the condition. In contrast, females with Rett syndrome can survive, and their lifespans are affected to a lesser degree. X-linked recessive inheritance involves a recessive gene on the X chromosome, where males with only one X chromosome will express the condition if they inherit the affected X, while females will need 2 copies of the recessive gene, one on each chromosome, to show the condition to its full extent. Colorblindness is an example of an X-linked recessive condition where males with one affected copy present with symptoms and females typically do not. Breaking it down in terms of inheritance, females who are carriers of X-linked recessive conditions may not show symptoms, but they can pass the condition onto their children. If they have a son, there's a 50% chance he will inherit the X and show the condition. If they have a daughter, she has a 50% chance of being a carrier, like her mother. Males who inherit an X-linked condition will always pass the affected X to their daughters. However, they will never pass the condition to their sons because they pass on their Y chromosome to male offspring, not their X. Understanding X-linked inheritance is crucial in predicting how genetic conditions might affect both females and males differently, especially when it comes to dominant versus recessive traits on the X chromosome. Mitochondrial inheritance is unique because it involves the genes in the mitochondria, which are the energy-producing structures inside our cells, often referred to as the powerhouses of the cell. Unlike most of our DNA, which is stored in the nucleus of the cell and inherited from both parents, the mitochondria has its own set of DNA, mitochondrial DNA, which is unique because it is only inherited from the mother. This means that only the egg contributes mitochondria and their DNA to the offspring, while the sperm contributes nuclear DNA, but not mitochondrial DNA. Mitochondrial disorders occur when there are mutations within the mitochondrial DNA. Mitochondrial DNA and disorders tend to affect tissues and organs with high energy demands, like the muscles, the brains, and the eyes. This is because the mitochondria are responsible for producing the cell's energy. In terms of inheritance, if a mother carries a mitochondrial disorder, she will pass it on to all of her children, but a father with the same condition will not pass it on to any of his children. So the only way to pass down the genetic information contained in the mitochondria is through the mother. Repeat disorders are another example of how our inheritance can be complicated. These disorders are caused by repeating DNA sequences copied many times within a gene. Imagine navigating a section of the maze that loops over and over on itself, making your journey longer and more challenging. Similarly, these repeated sequences can disrupt the normal function of a gene, affecting how it works in the body. In repeat disorders, the number of repeats can vary from person to person. For some, the repeats might be short and have little impact, but as the number of repeats increases, the effects can become more severe. This is because the longer the repeated sequence, the more likely it is to interfere with the gene's normal function, leading to problems in how the body works. In terms of inheritance, the number of repeats can increase across generations, often leading to more severe symptoms or earlier onset of the condition. This phenomenon is called anticipation. Fragile X syndrome is an example of a repeat disorder where an increase in the number of repeats in the FMR1 gene leads to intellectual disability. The severity of the condition often increases with each generation due to the expansion of these repeats. The fact that repeat disorders can change in size across generations is important when understanding inheritance. It can also make genetic counseling more challenging because of the risk of more repeats and more severe symptoms in future generations. You may have heard people say it runs in the family. By this, they are referring to the inheritance of genetic information from one generation to the next. This is how family patterns of traits or conditions are established. If a certain genetic trait like eye color or a genetic condition runs in the family, it's because the gene responsible for it has been passed down through multiple generations. However, not all genetic changes come from the family tree. Sometimes a new path appears in the maze that hasn't been walked before. This is what happens with de novo mutations. De novo mutations, sometimes called spontaneous mutations, are genetic changes that occur for the first time in a person, not inherited from either parent. It's completely random. There's no family history or pattern to follow. These mutations typically arise in the egg or sperm cells or early in embryo development. It's like finding a new route in the maze that no one has seen or used before. These spontaneous mutations can lead to genetic conditions that are not seen in previous generations. De novo mutations highlight the unpredictable nature of genetics, where new conditions can emerge without a family history. These conditions are particularly relevant when considering rare diseases, as many of these are the result of new, previously unrecognized mutations that cause unique genetic disorders. An example of a common de novo mutation that is a rare disease is Cri-du-Chat syndrome. We've now discussed the typical and not so typical inheritance patterns, but inheritance isn't just about the genes themselves. How these genes are expressed can be influenced by factors beyond the genetic code. This brings us to epigenetics and multifactorial inheritance, where changes in gene expression can occur without altering the underlying DNA sequence or changes that are affected by external factors. Let's dive into how these additional layers of gene regulation can impact inheritance. Epigenetics refers to modifications that affect gene expression without changing the underlying DNA sequence. These modifications could be things such as diet, stress, lifestyle, or even exposure to toxins, and they affect how the genes express themselves or how they work. Think of it like a maze with signs that guide you differently depending on the environment around you. The map may be set, but the signs along the way, which are how certain paths are marked, can shift based on various factors or exposures, like weather, for example. One key modification is DNA methylation, where small chemical tags, which are called methyl groups, are added to certain parts of the DNA, acting like switches to turn genes on or off. For example, environmental factors like diet, exercise, or stress can lead to changes in the pattern of DNA methylation, which can impact health by turning on or turning off specific genes associated with conditions like cancer, diabetes, or heart disease. Multifactorial inheritance involves conditions that arise from a combination of genetic and environmental factors. This means that while a person might inherit a genetic predisposition, environmental influences like stress, lifestyle, or exposure to toxins can affect whether or not a condition develops. For example, someone with a genetic predisposition to mental health disorder might not develop the condition unless certain environmental factors trigger it. In this case, genetics set the stage, but the environment influences the final outcome. Understanding these unique paths helps to eliminate why some diseases follow an unusual inheritance pattern that doesn't quite fit the traditional rules of dominant or recessive inheritance. It's an important factor to consider when tracing the genetic map in families, especially when dealing with rare conditions. Imprinting is a specific form of epigenetic modification where the expression of a gene depends on which parent it came from. So you inherit a gene, but its expression depends on whether it came from your mother or father. It's like having two possible signs for the same path, but depending on which direction you start from, one sign is chosen and the other is overwritten. So even if the genetic instructions are the same, the outcome can be different based on which parent's gene is active and turned on. In essence, the map of the maze might be identical, but the way the paths are marked or which paths are open and closed can change depending on whether the sign came from your mother or your father. For example, Prader-Willi syndrome and Angelman syndrome are caused by the same genetic region, but whether the condition manifests depends on whether the gene is inherited from the mother or the father. A related phenomenon is uniparental disomy, or also called UPD, which happens when a child inherits both copies of a chromosome from just one parent, rather than one copy from each parent. Normally, we inherit one copy of each chromosome from our mother and one from our father. But in UPD, this process is disrupted. This can lead to unexpected results in how genes are expressed, especially if there's a condition linked to imprinting. Imagine walking down a familiar path in the maze, but suddenly realizing that all of the signs have been replaced with the same direction. With UPD, this can happen when both copies of a gene come from the same parent, bypassing that usual balance that's needed for proper function. This can lead to conditions like Prader-Willi syndrome or Angelman syndrome, which arise depending on whether that extra chromosome came from the mother or the father. So while the map of the maze might seem familiar, the unexpected route taken by inherited both chromosomes from one parent instead of two can cause major changes in the journey. These mechanisms highlight that gene expression can be influenced not just by genetic sequence, but also how genes are regulated. Epigenetics opens the door to potential therapeutic strategies that target gene expression. By understanding how genes are switched on or off, our researchers can develop treatments that might flip the switch in the right direction, offering potential solutions for conditions caused by that improper gene expression. Lastly, we have mosaicism. And mosaicism occurs when some of a person's cells carry a genetic mutation while others do not. This means that the person may have different genetic information in different parts of their body. For example, a person with mosaic Down syndrome may have some cells with the typical 46 chromosomes and others with an extra chromosome 21. The resulting condition can vary in severity depending on how many of our cells are affected. Mosaicism can make diagnosis more challenging, as the genetic change may not be present in every cell, leading to variable symptoms or traits across the body in different parts. If you're interested in learning more information regarding these very complicated and unique inheritance patterns, please take a look at all the links we provided at the end of the video. To understand where you're going in the maze, or if there are things that run in the family, it helps to have a pedigree. This is like a family tree that shows the genetic paths taken by your relatives over generations, allowing you to see patterns that might not be immediately obvious. Genetic counselors use this information to look for patterns in how certain conditions are passed down. They can spot whether a condition might be inherited in a dominant, recessive, or other pattern, and they can help predict the likelihood that you or your children might be affected by certain genetic conditions. Having a family medical history is essential for this process because it can reveal potential risks and help shape decisions about genetic testing, management, and prevention. While not everyone knows the full map of their family's history, and not every family talks about their medical health with each other, it can still be helpful to gather as much information as you're comfortable with. While taking your family history, genetic counselors might ask you about two different things that will help to inform their assessment: ethnicity and consanguinity. Another important aspect of understanding your genetic map is knowing your ethnicity. Certain genetic conditions are more common in specific ethnic groups, which means ethnicity can provide valuable context for understanding potential risks. For example, sickle cell disease is more common in people of African, Mediterranean, or Middle Eastern descent, while cystic fibrosis is more prevalent in people of Caucasian or Northern European descent. By knowing your ethnic background, a genetic counselor can help identify whether you might be at a higher risk for certain conditions that are more common in particular populations. This is especially useful in carrier screening, which tests whether you carry a gene for certain conditions, even if you don't show symptoms. A person can be a carrier of a recessive condition like sickle cell disease or cystic fibrosis without knowing it. Carriers usually don't have the condition themselves, but they could pass the gene on to their children. If both parents are carriers of the same recessive condition, their child has a higher chance of inheriting the condition. So understanding ethnic background can guide which tests to offer and whether you need to be tested for specific conditions. We may also ask about consanguinity, which refers to whether there's a family history of related individuals, such as cousins, having children together. This is relevant because consanguinity can increase the likelihood of inheriting genetic conditions that are recessive or otherwise rare. In populations where people have a shared ancestry, genetic traits can become more concentrated, and the risk of inheriting genetic conditions can arise. It's important to note that consanguinity doesn't automatically mean that a child will have a genetic condition, but it can increase the risk of certain conditions that are more common in closely related individuals. In these situations, knowing about consanguinity can help genetic counselors recommend appropriate testing, preventative measures, and family planning options to help families make informed decisions about their health. Knowing these patterns about your family's genetic history, including ethnicity and consanguinity, is essential for genetic counseling and understanding your risks. It's not just about predicting conditions, it's about empowering you with knowledge to make decisions about your health, future family planning, and even lifestyle choices. It's about knowing the genetic paths you might encounter, the ones you might want to avoid, and the ones you can confidently navigate. Now that you have a better understanding of how genetics shapes your path, it's important to think about how you can explore that map more thoroughly through genetic testing. Genetic testing helps uncover hidden routes, identifying risks and conditions that you may not have been aware of. It's like examining the map in detail to see which paths might be more challenging or where obstacles may arise in your journey. From carrier screening to prenatal testing, there are various ways to gain insights into your genetic landscape and make informed decisions about your health and your family's future. However, sometimes discussing genetic testing and its implications can be difficult, especially if there are concerns about whether a condition runs in your family. It can feel like you're facing an unknown or unsettling part of the maze. This is a completely normal feeling. Conversations around genetics, whether it's learning about a family history of conditions or considering testing for carrier status, can be emotional and complex. You may worry about what the results will mean, how they'll impact you or your loved ones, or how to explain it to others. The good news is you don't have to navigate this part of your journey alone. There are many resources available to you. A genetic counselor can help guide you through these conversations, offering expertise on testing options, explaining what the results might mean, and helping you understand how to communicate these findings with your family. Genetic counselors are trained to make these discussions easier, breaking down complex genetic information and helping you make decisions based on your personal circumstances and values. In addition to professional support, there are also support groups and community resources where you can connect with others who may be going through similar experiences. Talking with people who understand what you're going through can provide comfort and perspective and can help you feel less alone in the process. Many organizations provide tools and advice on how to talk to family members about genetic health concerns in a way that is both sensitive and constructive. Whether you're exploring genetic testing out of curiosity for a specific health concern or because you're planning for the future, these resources can empower you to take the next step with confidence. Your genetic counselor can help guide you through the maze, ensuring that you have the information and support you need to make informed, thoughtful decisions about your health and your family's future. So don't hesitate to reach out and explore your options. There's always help available as you navigate your unique genetic journey. Remember, navigating the maze of genetics isn't always easy, but with knowledge, support, and the right tools, you can better understand your genetic journey. And just like any maze, you don't have to walk through it alone. There are genetic counselors, support groups, and resources that can guide you every step of the way. Thank you for joining us on this genetic adventure. To learn more, visit nsgc.org or connect with a genetic counselor today to find one through this website. Keep exploring, keep asking questions, and remember the maze is yours to navigate, as well as click on the links that we have listed or look into more of our videos to learn more information.